Welcome, everyone. Thank you for joining today's webinar. My name is Melissa Zemik, and I'm the Communications Advisor for Applied Research at Olds College. Today's presentation is called Remediation of Contaminated Water from a Livestock Farm using Floating Island Technology and Native Wetland Plants. Before we begin, we want to acknowledge the funding and technical support from Tanis Conservation Services Limited, Alberta Real Estate Foundation, GP Restoration Solutions, and Highfield Investment Group for the successful completion of this research study. And we'd like to also thank RDAR and UFA's support for the next step of research. Your support to Olds College is all greatly appreciated. And just a quick overview of the webinar. Patty Morris, Executive Director of Alberta Real Estate Foundation, our project sponsor, will be saying a few words about their foundation and sponsorship. Joy Agnew, Associate VP of Applied Research at Olds College. Olds College Center for Innovation will be giving a quick introduction to applied research at the college. Stephen Tanis, President of Tanis Conservation Services and the lead scientist of this project, will be giving the main presentation. Dan Karen, faculty instructor and research scientist with Workland School of Agriculture Technology at Olds College, will discuss future research of floating island technology. And for questions, we saved time at the end of the presentation. So if you have any questions during the presentation, please type them at any time into the Q&A box in your Zoom control panel, and we'll go through them at the end. Or during the question portion, you can simply raise a hand via the control panel or unmute yourself just to ask it yourself. So I'd like to introduce Patty Morris, the Executive Director of Alberta Real Estate Foundation, to say a few words.
Thanks so much, Melissa. Hi, I'm Patty Morris, Executive Director of the Alberta Real Estate Foundation. And please let me begin by thanking Olds College team and researchers for inviting us to celebrate the impact and findings of phase 2 of this important work on floating island technology. Could I have the next slide, please? For those who we haven't met yet, we're really excited to have the opportunity just to quickly introduce ourselves. Alberta's very unique. We're one of 2 provinces whereby under the Alberta Real Estate Act, when a consumer deposits money in trust through a real estate transaction, either a residential or a commercial transaction, the small interest that's earned on that is accumulated by our foundation and it's reinvested in Alberta communities. Next slide, please. This is our 30th anniversary, and since 1991, we've had the privilege of investing $26.5 million in grants to over 665 initiatives. Next slide, please. Under the governance of our board and guided by the Alberta Real Estate Act, the foundation identifies and invests in projects that align with our legislative purposes, and we hope and contribute to positive outcomes for industry and our communities. Next slide, please. Through educational initiatives, law reform, regulatory improvement, research, and the development of land management and stewardship practices, the foundation aims to contribute to thriving Alberta communities. Next slide, please. We've been really honored to support phase 1 and 2 of this research. And as of last week at our board meeting, we're excited to continue to support the project funding of phase 3. And for us, this conversation today is so timely. As part of our 30th anniversary celebration, we've been hosting a number of stakeholder engagement events, which have really been intended for us to hear from our stakeholders and ultimately to use their input to help chart our future. And one of the things that we heard resoundingly from the industry was the massive changes that the real estate industry is facing, including demographic, cultural, social, financial, and environmental. And critical amongst this was water health, protection and enhancement of property values through better land use planning and management, and of course, the devastating impacts of climate change. As a result, we're continuing to explore how we can support efforts that help industry keep pace and adapt to these changes. And a great example of that for us is this research taking place at Olds College on phytoremediation, which we think holds great promise for use in Alberta and beyond. So we look forward to learning more about the important research findings. And again, thank you for including us in this, this opportunity. Next slide, please. And just thank you again. We always welcome you to stay in touch if you're aware you possibly have a grant idea yourself or just want to learn more about the foundation. please don't ever hesitate to reach out. We really look forward to those opportunities. Our door is always open. Thanks so much.
Thank you, Patty. Here is Joy Agnew, Associate VP of Applied Research at Olds College Center for Innovation.
Thanks, Melissa, and thank you to everybody in attendance, and thanks, Patty, for that warm intro and warm welcome. Really quickly, I just wanted to introduce or highlight the Olds College Smart Farm and some of the work that we're doing with applied research at Olds College. So for those of you that don't know, Olds College's farm operations, including 2,800 acres of land, a cow-calf herd, a feedlot, and a sheep flock, are essentially operating as closely as possible to a commercial farm while giving us the flexibility to operate it like a living lab for enhanced teaching and learning opportunities and applied research, all with a focus of innovating, developing, validating, and demonstrating and scaling new technologies, processes, ideas, and philosophies for the betterment of ag and food production. Next slide, please. The Olds College Center for Innovation is the research division of the college. And so we leverage and utilize the smart farm operations and other infrastructure and assets of the college to conduct applied research, all with the focus of accelerating the development and adoption of technologies and practices to improve productivity, profitability, and sustainability of ag and the agri-food industry, all through industry-driven applied research. So this project specifically is a wonderful example of leveraging multiple experts across our sectors, including livestock research, environmental stewardship, and our technology integration research team to tackle a problem with strong industry push and pull. So we have innovators and small to medium-sized enterprises on one side of the equation, and end users and real estate foundation and the livestock industry on the other side, all benefiting from the work that is happening with a project like this, evaluating the potential of native wetland plants for management of water. Next slide. So a quick outline, and I apologize for all the words on the slide, but I didn't want to miss some of the important details about how this project came to be. Because as Patty outlined, we're now just starting phase 3 of this initiative. So throughout the 2010s, essentially, Olds College built capacity, infrastructure, and expertise related to the evaluation of constructed and natural wetlands. And so through a partnership with a local innovator, TCS Limited, and other industry partners, and under the guidance of our now retired research technician, Ruth Elvestad, we were able to secure funding for a multi-year initiative. Phase 1 started back in 2018 and involved controlled greenhouse trials and water spiked with nutrients. And we hosted a webinar outlining the research findings from that phase of the work about a year ago, and it is available on our website. Phase 2 was initiated in 2019 and continued the controlled greenhouse trials, but this time used water from the Olds College feedlot and expanded the different plant species that were evaluated. And that is going to be the focus of today's webinar, are some of the findings from the phase 2 research. Phase 3 just launched this year and involves a pilot scale evaluation at multiple feedlot sites in central Alberta. And that phase of the research will also be outlined, or the plans for that research will be outlined today as well. The ultimate goal of this multi-phase initiative is to evaluate the technical feasibility and economic viability of using floating islands and native wetland plants to treat agricultural and industrial water to ensure the water poses no risk for contamination and can be recycled for irrigation or as a livestock water source. I just wanted to acknowledge the fact that Alberta Real Estate Foundation and Highfill Investment Group have been behind us from the beginning, right from phase 1 of this work. And I just wanted to heartily acknowledge that and thank them for that ongoing support, as well as ARDAR and UFA, who have come on board to support this very important pilot work as part of phase 3. So with that, I'll turn it over to the scientists and the experts to tell you all about what we have learned in phase 2 and what we're hoping to accomplish in phase 3. Thank you.
So now Stephen Tanis is going to be taking over control of the screen and presenting on his research from phase 2. And don't forget to unmute yourself, Stephen.
Sorry, everything jumped around my screen when I set up there. There we go. Yeah. Good morning, everyone. Thank you for coming and to attend this webinar. And yeah, I'm happy to present some of the research we found over the last number of years. I will be touching on phase 1 and then focusing on phase 2 so that you'll get the grasp of the depth of where we've come from and where we're going, and let Daniel take it into the future as we move on. So as an introduction to this presentation, my company is Tanis Conservation Services. We are a small company, less than 15 people in the Cremona area. We've been operating for the last 35 years under my parents and myself. And we are a specialty service company in the environmental field: reclamation, phytoremediation, restoration ecology. We really focus on challenges, and we like to do a lot of research on programs where people have challenges that are limiting what they can do. And so we look for those areas where we can help and develop new technologies to solve problems. We do this in biophysical. We teach around 200 to 300 people on average a year in professional services. And those courses are in botany, bioengineering, reclamation, wetlands, so a number of different areas. And then we have started a second company that is focused in on product manufacturing, and that's floating islands, native seed harvesting equipment. And this is all built in Canada and basically focusing on creating products that are specialty to our climate. And we do sell now worldwide to 44 countries with our harvesters. And the islands are patented, and hopefully we'll be starting to move that process forward. So as we get into the actual experiment, what is the problem that we have? Well, the problem is contaminated water. And this is a global problem. It's continually getting worse, where we are with higher densities of populations, we have higher pressure on our ecosystem. And what happens is we end up with situations where you have heavy metal loading, we have salts, we have hydrocarbons, and one of the main ones is eutrophication, which is nutrient loading into water systems. And the problem with this specifically is that it leads to some very, very dangerous outcomes. Algae blooms, which many people have heard about, lots of beaches being closed down because of algae blooms. When you get a lot of nutrients, something is going to start growing with all those nutrients, and a lot of times it's something you don't want. And so algae blooms reduce the water quality, deplete oxygen levels, and lead to undesirable smells and aesthetics, but they can also be dangerous as well, depending on the algae blooming. This is really led by some of our main nutrients: phosphorus, nitrogen. So those macronutrients we use in fertilization, in agricultural lands, golf courses, industrial applications, and commercial and residential uses. And you might not think of like mining, but they use in their blasting, nitrogen is released. So this really spans all industry, and agriculture, being one of the largest footprints on our landscape, becomes one of the areas with the largest issues around eutrophication. So it is an area that we are concerned. I will touch on a few other areas that I've researched, but plants specifically, we know, use nutrients. Can we get them? So phytoremediation, can we get the plants to actually be harnessed to actually remove the nutrients we do not want? And if you haven't seen an algae bloom, when you see this green on the water, that's where there is duckweed and there's some other things that can be, but you watch for that green on the water, and that's where you're going to see the algae blooms. Sometimes it's other colors as well, depending on what algae you're dealing with. So we do know that plants will uptake nutrients. That is why we fertilize. Wetland systems are known for being water filters. So the plants that grow in these systems are adapted to high nutrient situations. Many times we have nutrient loving plants in wetlands. And so selecting in wetland systems for plants that take up these nutrients is a perfect place to start for nutrient removal. So we're in a situation that this has potential. In the past, it has been found that nitrogen has been effectively removed, phosphorus. Selenium is an area that I've done for the last 6 years in mining industry, and we are getting 35% removal of selenium. We won't talk about in this presentation the details of that, but we are getting very, very good removal in selenium. Sulfates, there's research happening, Dr. Finley in Saskatchewan on sulfate right now, and looking at dugouts for that specific research. So we have research happening in other areas here in Canada as well. And potassium is another one where there's been good results for removal. So artificial platforms, what are floating islands? Basically, it's something that can grow plants on water. We don't have the ability to grow in many of our water bodies plants on the shorelines because of the water fluctuation levels. So many stormwater ponds, settling ponds, your water levels drop by a meter and then they'll rise by a meter. Most plants are adapted to 10, 15 centimeter fluctuation zones. Our bigger cattails, bulrushes can take 50 centimeters, 80 centimeter shifts. But when you start getting meter, 2 meters, 3 meters, basically the plants will dry out and die, or they'll get flooded out and drowned. So we need something that can adapt to that situation. So floating islands allow us for versatility in our installations. So when you can't use a shoreline planting, you can use them. They are really good in fluctuating water levels. They increase the surface area, so shorelines aren't enough. We can actually put plants on the water to treat, and can be retrofitted on pretty much any installation. So in the pictures down on the side here, the top, we have Lethbridge County, some little islands that they are playing around with, that they were donated. We have Olds College in the middle here, some of our research islands, and then Lethbridge, the city of Lethbridge, this is their installation of floating islands that are on one of their stormwater ponds. So there are limits though in floating islands. There's a lot of research that's happened in them, so we do understand some of the limits. We have had problems with some of the commercial islands sinking. You have to make sure you're buying a product that has good flotation. If it's a sample bottle, if it's some of those type of things that can't withstand ice and long-term flotation, then it's not going to last in our climate. Maybe good in some other climates, but it may not last in our climate. Many islands are built without the needs of plants in mind. They'll create something that floats really well, but they aren't thinking about a plant. So foam islands breaking apart due to wildlife. We've had really a famous one. We've had at Olds in a number of areas where some of the foam islands require irrigation. And if you're irrigating your islands, that's kind of defeating the purpose. If you're on water, you should be able to pull the water from the actual water body. So there are some islands where the plants are dying because they need irrigation. So we have had challenges. That's where we have gone to developing some very specific islands that deal with these type of situations. The photo you see on the side, this is after herbivory. The plants, we put our test island out in the pond, and ducks and geese came in and just ate everything off. So we have to have protection for some of these little fences that we have on the side. The island on the bottom there, this is one that is just coming out of spring. It froze down, the water, the ice sank to the bottom, where water formed on the top of the ice, and it was underwater for a month, and it just, as soon as the ice melted, it popped up to the surface. That's what we're looking for in durability. And these are the considerations in this process that we're dealing with. Research into plants, though, has been limited. Many people have studied cattail worldwide, but that's about it. But cattail does not grow in all situations, even though it's a very good growing species. Low nutrient conditions, as we'll talk about later, really affects cattail. So there's problems with what we know about our species. And we don't want to bring in weeds, bring in flowering rush, Phragmites australis, really well-known species out of other parts of the world. They're regulated weeds here. So we do have to think about that, and we have to understand what each plant does. So in AREF1, the first experiment, we used native plants, a number of species, grew them in the greenhouse in tanks, and then spiked the tanks with a hydroponic fertilizer solution. So provided everything with distilled water, very controlled situation. Cattails stored more nitrogen, phosphorus, and potassium and sulfur in their shoots than any other species. And the growth rate on these plants, this is photo beside is from the second round. The first round, the plants, I think were 12 to 14 feet, somewhere in there, the cattail. They got huge. And it was driven by the nutrient regime. The cattails were very, very effective at pulling down nutrients. Lowest concentrations of phosphorus, potassium, sulfate remaining in the water, and the highest evapotranspiration rate. If you're trying to get rid of water, the cattails remove 10 times the water of evaporation alone in these high nutrient conditions. This changes when your nutrient levels drop. Nitrate concentrations did not differ from the controls in any of the species other than cattail. So basically, the cattail was the only one in nitrogen that showed the really significant drops in nitrogen. Phosphate concentrations remained in plant treated water were higher than the control, but we had an algae bloom, and that caused a lot of havoc in our analysis. And that's something we corrected in the second round. Potassium dropped below control for all plant treatments. So we had significant removal of potassium in all plants. And then sulfate was lowered in all species other than water sedge. So for AREF2, we wanted to determine the effectiveness of 7 species. So looking at a little broader group of species, dropping the worst performers and looking at more broadly. We wanted to determine the water use efficiency. We hadn't thought of that before. We realized how much water we were having to add to these tanks weekly, or they would dry out. And then determine the potential of each species to be used in a floating island system, grown in water, not rooted on a shoreline, which is very important. And then test the constructed wetlands using with feedlot effluent. So we're looking at real world situation water now. And so this means it affects what we're actually testing. So we were working in a batch mesocosm, this style of experiment. The custom-made islands were built just for these specific tanks. They're not a long-term outdoor island. These can't take freeze thaw as much as other islands, but they are a solution for a shorter term. They are 46 by 71 centimeters with 7.5 centimeters of peat moss as our growing medium. So we looked for a natural growing medium that had microbial capability to function as well. So we've got the fully functioning system here with microbial interaction with the plants. You don't want to eliminate microbial because that is an important uptake for many plant species. We have 5 plants planted per island, 24 tanks with 3 control tanks, and all tanks were aerated and bubbled every 20 minutes every 4 hours. So we're looking at an aerobic system. Plants installed November 19th, 2019, grown until July 13th. We were supposed to be starting up after about 4 or 5 months of grow out. The idea is we put in little plugs, as you can see in the picture. We grow them out to their more mature plants, and then we run the experiment on mature plants. The issue was COVID hit, and so we delayed our startup, which shortened our treatment time, which I'll explain a little later. Basically, the plants have been growing for a year and decided naturally they wanted to senesce out. And so we had them just naturally senescing at the end of October. That kind of finished off the experiment. Our species, we have Carex 30, so wheat sedge, smartweed, common cattail, mare's tail, water sedge, Baltic rush, and small fruited bulrush. Looking for biomass producing plants that can have a different environmental conditions than they withstand. The water was used from the Old College feedlot holding pond. We added July 14th or July 13th and sampled July 14th, so 1 day shift in time, and you'll see what happened with 1 day here. And we looked at nutrients and salts and metals. And then with vegetation, we sampled it before starting and then at the very end, looking at the roots and the shoots at the end of the results. So 1 day after we sampled the tanks, nitrate levels were low in the water. So we were below detection limit, but we didn't sample all nitrogen levels. And we will be doing that in the next round. It was something to do with the lab and what we got. What we did find was in one day, you see the drops in our nutrients. So from July 14th, this is July 14th, the water that was added. So the direct water we added, this is one day later in the tanks, we saw drops immediately from the controlled water. Cattail did not perform as well. That is something that was clear in this, and that nitrogen is likely a big part of that. When we looked at biomass productivity, cattail in the first experiment was by far above everything else. And in this one, it's one of the lower, it's the second lowest in biomass in wet and dry weights. So definitely something that you have to think about. Or then small fruited bulrush though was a very good biomass producer, followed with Baltic rush, which surprised me a little bit. I was expecting the two sedges that came in next to be higher than Baltic rush, but Baltic rush is a nitrogen fixer. And so in a low nutrient environment, it might be the key species to be using. So if you have low nitrogen but high phosphorus, Baltic rush is likely one of your key species. Looking down at smartweed, mare's tail, smartweed just didn't do as well in the islands, specifically to our environment. Smartweed can be one of the biggest plants in the experiment. So that's something that I might have to look at in the future, is how to get it to grow properly. What we saw for plant growth is, you can see there's a trend here. The tillering is how many leaf units are coming out of the plants. And as we did the counts, you can see these plants were continuing to grow. The only ones that really didn't was our smartweed and cattail. They leveled off and did not produce more, where the other species were all on a growth trend, and even to the end of the experiment. So we know that as time goes on even further, they are going to continue growing. So what we found, phosphorus, this is the water results. And we sampled the water every 4 weeks, on average 4 to 6 weeks. And what you can see is the drop off from the beginning, July 14th, the red dotted line is the control. And you can see we had a consistent with all the species phosphorus rates dropping. So that's really important. Wheat sedge, your Baltic rush, there's a number of those species that performed very well. Pondweed, which did not get the biomass production up, struggled. So that was the only species that really struggled. When we looked in the concentration, so where you want your biomass is in the shoot. So I'm focusing on this presentation on the shoot biomass, on what concentration we had. And we found that the wheat sedge had the highest concentration. Pondweed actually had a high concentration as well. So if we can get biomass production up on it, it would be a good species. But water sedge as well had a high, and the small fruited bulrush. Those three kind of had the highest, followed by Baltic rush and mare's tail, and cattail with the lowest. Again, cattail without its nitrogen was struggling. When we look in the soils, so this is what's remaining in the soils, and it's important we sample every component. We found that pondweed had high phosphorus in the soils. So it wasn't going into the plant and it ended up in the soil. But looking against the control, the phosphorus in soils was the same as control for the water sedge and Baltic rush. So they didn't have elevating in the soils. A few of the other ones had a little bit of elevation, and that might have been with some of the microbial biomass that was developing in there. So nitrogen in the shoots. Well, we couldn't do nitrogen in water because it was below detection limit, but we could look at what's happening in the shoots. Baltic rush had the highest in the shoot, again, nitrogen fixer. So it was able to solve its own problem here with low nitrogen. So not surprising, it was the highest concentrations. Water sedge, wheat sedge, and then mare's tail were the next ones on the list. And I have to do some work with some of these species, but mare's tail had a lot of potential in pulling nitrogen. So did wheat sedge, which we had seen from the previous iteration of the study, where pondweed and cattail did not have much nitrogen in them. In the soils, when we compared to control, only pondweed had elevated nitrogen. Everything else was reduced below in the soils. This was a nitrogen deficient system, and they were pulling out the nitrogen as much as they could in the system. Okay, then potassium, what did we see here? We had the control, you can see is at the very top here. Everything was drawn down in potassium. Poorest performers was pondweed and water sedge, but the best performers in potassium removal were in the cattail, mare's tail, small fruited bulrush. So we have a number of those species that were really, really effective in that pull down rate in the water. The concentrations in shoots, again, we wanted it to be in the shoot, not the root. And I do have shoot to root ratio information as well, but cattail had a lot in the shoots. Water sedge did okay. Looking at mare's tail, it was a really good performer here. Wheat sedge had lower in the shoots, and small fruit bulrush fairly good in the shoots, pondweed as well. And when we look at the concentration now, it's the biomass by the concentration is what gives us the final result on the best performers. And so our big performers, such as small fruited bulrush, the water sedge and wheat sedge were the big biomass producers, and that can affect where they would be. In the soils, potassium, the control you can see is up high where all the others were dropped. So they were taking the potassium out of the soil as well as the water. Sulfate, we had something interesting happen, and this is the one that we used tap water in the experiment, and we think that there was some sulfates in the tap water, and as we're watering continuously, we're adding to the system. Also, there's a potential that our peat had sulfate in it, and as we're breaking down the peat with microbial, we're releasing sulfate there as well. But what basically happened was with our control, you can see it has elevated even. So even the control was elevated sulfates. The plant species with the highest water uses and vegetation growth generally trended to being the ones with the highest sulfate, which we believe is because we were adding sulfate to the system more frequently to keep the water levels up for those species. So that is something to be considering always. If you're adding water, the biomass of like the volume of water we were adding was at such levels that we couldn't keep doing it as distilled. It was just unmanageable from the first experiment. So this is where you can see there was a drawback there. In vegetation, mare's tail held the highest sulfate. So this is an important finding, and small fruited bulrush had this second highest concentration. So that is really important information that we did gain. Is there were a couple of species that clearly outperformed. Small fruited bulrush has a lot of biomass production as well. Mare's tail has a moderate biomass production. So that is something to consider. In the end, those two might be fairly equal in the actual net result of removal. So water use. This is something that you can see in July, August, we had the highest water uses. You can see the control is way below. That's evaporation water use and what we had to add to the tanks. Again, the species that had the poorest growth, like pondweed, had also the lowest requirements for water. Mare's tail, for being a fairly productive species and effective, had low water use. So if you're in a system where you don't want to remove your water, this is important to select species with lower water use. If you're in a system where you want to remove water, then you want to use species that use more water. Cattail fell off. It did really well and then started to drop off. And what it was is it could perform for a while, used up its nitrogen, and my theory is that's why it started dropping off, where in the other experiment, it just continued to go up. So important to know your system. For plants, we found nutrient deficiencies may limit growth. Cattail, water sedge, wheat sedge had the highest plant height in tillers, and they were almost double in trial 1 with high nutrients versus trial 2. During senescence, nutrient removal drops, and we stopped the project because we had, I'll show a picture later here, the plants were just starting to senesce off. It was the end of the season and they could not do anymore. So for phosphorus, we removed phosphorus effectively when we started doing the analysis. Removal was between 77 and 84% of our actual phosphorus was out of the water. This is above what some of the other meta-analysis have found in the past. And we did not have the algae bloom problem that we had in the first round. So we were able to solve that and drive the phosphorus into the plants. Phosphorus storage locations differ in species. And so we found wheat sedge, water sedge, smartweed, and small fruited bulrush kept it most of it in the shoots, and that's really important for harvesting them off and removing phosphorus from the system. And then we want to know the biomass production. That's the other part of the formula we will be focusing in on next. Nitrate removals from water is key. And historically, about a 58% removal rate has been found. We weren't able to detect nitrogen, so we weren't in a system where we could do that in this situation. But it's something to know that we can remove by past research. And our last round of studies found that we were effectively removing nitrogen. Next time we will test more versions of nitrogen to counteract this issue. Let's make sure there wasn't nitrogen there we weren't testing for. And high oxygen levels can reduce nitrogen removal, and that's something that we'll be considering in the next round, where we are not going to be oxygenating at the same levels as we were here. For potassium, the experiment showed a 27 to 45% removal depending on species. Cattail, mare's tail, small fruited bulrush, and Baltic rush, water sedge, and weed sedge all were effective. So we had a lot of species effective in this. And the plant material can degrade when it's senescing, and you can lose your potassium back to the system. So you need to consider whether or not removing the above-ground biomass, or if it's being defoliated naturally by birds, that may counteract and help you. So thinking about defoliation is important here. Sulfates increased over the experiment, and this we think was the tap water and soil decomposition may have been related to this. And so those considerations are going to be playing out in, we didn't find sulfate increases last round where we used distilled water. So the tap water may have been the big culprit. So for our future, what we're going to be doing is assessing the wintering effects in cold climates, and looking at these in actual actual outdoor situations. We are doing it on mining facilities already. Testing for ammonia, ammonium, total nitrogen, all the forms of nitrogen. That is a key improvement. Count for biomass in nutrient accumulation. We did a really good analysis on this in phase 1. And so in phase 3, that'll be a goal. And increasing nutrient loading to more typical feedlot water. So the next round, we are going to the field to actually test it there. And that is the end of my presentation. I'll pass it to Daniel.
Right, can everyone see my screen?
Yes, you're good, Daniel.
Okay, thanks, Stephen, Joy, Patty, and Melissa. Good morning, everyone. My name is Dan Karen. I'm an instructor at Olds College, and I'm the lead scientist on phase 3 of this research. And so phase 3 of this research is an ARDAR and Alberta real estate funded project, which brings the floating islands into real world situations. Where the first 2 phases were controlled environments, now we are installing the islands and measuring their performance in commercial feedlot holding ponds. We will monitor a wide variety of water quality parameters, but like before, we are particularly interested in nutrient concentrations. And this time we are also interested in fecal indicator bacteria, total dissolved solids, and salinity. Our overall objective is to provide feedlot operators access to alternative low-cost options for treating feedlot runoff, with the hopes that the treated water will be more suitable for reuse for either livestock watering or irrigation. And so, because we are now testing these islands in uncontrolled environments, the research becomes significantly more complex. Feedlot holding ponds are very complex environments where water quality parameters and the availability of different contaminants are in a constant flux. And so recognizing that, we need to collect diverse data sets with an appropriate experimental design. The design we chose is called a before-after control impact design, or BACI for short. And BACI designs are robust experimental designs used frequently in ecological studies, which is essentially what these feedlot holding ponds are: diverse ecosystems of microbes, plants, and other organisms. And so with this design, we will study 4 feedlot holding ponds, where 2 of the ponds will have islands installed on approximately 5% of the pond's surface area. And then the other 2 ponds will be used as controls. In our first season, all of the ponds will be monitored without floating islands, giving us the before impact. And then in the fall of 2022, next year, we will install the islands on the 2 ponds. Then during the following season, the 2 ponds with the islands will be monitored for the after impact, and the 2 control ponds will be monitored for the control impact. And so for the 4 study locations, one of the sites is going to be the Olds College feedlot, and the other 3 sites are provided by our 2 partners, Clausen AgriVentures Limited and Penridge Feeders Incorporated. We are really grateful to be working with these 2 great producers who are conveniently located just east of Linden, within 20 kilometers or so of each other. Ponds at the study location range in size from 0.1 hectares to 0.67 hectares. The Olds College and the Clausen AgriVentures West sites will be the controls in this study. And Penridge Feeders will have 14 islands installed next fall, and Clausen AgriVentures East will have 41 islands installed next fall. And so the water in this project will be analyzed for a wide variety of parameters. First, water samples will be collected bi-weekly from each pond during the season. We also have automated samplers that will be set up to capture runoff samples during rain events, so we know what kind of contaminant loads are entering the ponds. Soil and plant tissue samples will be collected at the end of each season for lab analysis, like in the previous phases. Deployable multi-parameter sondes will be deployed in each pond to provide a continuous data set of physical and chemical pond dynamics, including parameters such as temperature, pH, dissolved oxygen, oxidation reduction potential, and total dissolved solids. And so, since the goal of this project is to improve water quality for potential reuse by producers, we are particularly interested in all the things that make the water unsuitable for that end goal. Nutrients are of interest because they are the underlying cause of algae blooms, and especially blue-green algae blooms, which can release harmful toxins. Learning our lessons from the previous phases, we will be collecting all forms of nitrogen and phosphorus so we can really characterize how nutrients are cycling within these environments. We're also measuring the amount of algae biomass being produced with chlorophyll A sensors connected to the sondes. And this should help us understand how productive these environments are and how the floating islands will compete for nutrients with the other aquatic plant life. Fecal indicator bacteria are also of interest because they represent the presence of pathogens, which are harmful to humans and some animals. And then finally, total dissolved solids and salinity are of interest because livestock will actually refuse to drink water where the TDS is too high. And saline water that's used for irrigation is destructive to field crops and to the soil. And so understanding contaminant concentrations is important, but to really interpret them in a meaningful way, we need to know how much water there is. And so with this project, we are measuring the quantity of feedlot runoff that is entering the pond, and we're measuring the pond volume with bathymetry data and water level loggers. There are no outlets on any of the ponds, and they are all lined with clay liners, preventing groundwater seepage. Thus, we only expect our only output is going to be evaporation, and we will calculate that with climate data from the weather stations installed at each site. And so I would like to also take this opportunity to give a special shout out to Alberta Agriculture and Forestry for lending us the H-flumes and automated samplers for this project. We are greatly appreciative. And so just to give you all a brief timeline, by next spring, we expect to have all of the sites instrumented. After that, we will monitor all of the ponds without the floating islands. Fall of next year, we will install the floating islands on 2 of the sites at Penridge Feeders and Clausen AgriVentures East. We will process our results for season 1 the following winter. During season 2, which is April to October 2023, we will monitor the control ponds and the ponds with the islands installed. And then in the winter of 2023-24, we will process our final results and analysis. And so that's the plan for phase 3. I would like to finish by thanking our current partners and potential partners who have shown interest. We continue to engage with and establish partnerships with industry and other researchers to enhance the outcomes from this work. So please get in touch if you would like more information or have ideas for potential collaborations. And that's it. I think we can move into questions. Thank you.
Yes, we have actually quite a few questions. And thank you. So that concludes the presentation. And we're going to go ahead and take questions from the audience now. And please keep in mind that I'm not a researcher. So if I mispronounce some of these words in the questions, don't laugh at me. So Dr. Tanis, question 1, how was the algae bloom in the control treatments resolved in phase 2?
So one of the main things we did in phase 2 was resetting our islands to shade the water surface. So we didn't provide surface water area for the algae bloom was one of the big ones. We debated a bunch of different treatments, but we didn't want to kill our microbial biomass. And I can't remember if we did something else, but I believe the main thing was actually just working on our shading, to shade out with the island, the water surface. And if anybody else has any other information on there, but that was the main thing we were doing.
Okay, and next one, Dr. Tanis, where did Baltic Rush fix its nitrogen from? The air, question mark?
So Baltic Rush, it is coming from, it has nodules on the root system. So it's usually coming from through water and air, but it's the microbial association, like just like your alfalfa, your nitrogen fixing plants. So it is coming eventually from the air, but it is, yeah, in the water as well.
Okay, and next question, how did the student researchers support the work you did? What kinds of activities did they do?
Student researchers. So we had a lot of, we had water sampling, we had tiller heights that had to be accounted, and that's a lot of work. Tiller numbers. So a lot of the daily sampling of and weekly sampling that we did throughout the process, that's where we got a lot of those supporting help.
And Dan, I think you can add to that as well.
Yeah, so the students have been actively involved with phase 3 of this project as well. So we've got a directed field study student from the Bachelor of Science in Agriculture program that is helping us with sampling. Her name is Emily Edgar, and she's helping the research technician with sampling, the installation of all of the equipment, and she's going to be helping us with some of the analysis as well.
Perfect. And next question, why were you adding sulfate in the water? What was the rationale?
So we weren't actually intentionally adding sulfate. We were using tap water to actually add water to the system, because these plants are pulling down the water in their tanks weekly and could drain the tank of water on basically a weekly or every 2 weeks basis. So we have to add water to the tanks, ideally use distilled water, but because of the volume of water to distill it, we end up using tap water in this, and that's where we think it was coming in. So unintentional.
And next question, do you think the slow, poor uptake of nutrients by smartweed shoots might be mitigated longer term, since they did have large amounts in soil with microbial connections? Maybe those soil aspects would be important longer term. And if it is in the soil, isn't it still not in the pond water, which is a good thing?
Yeah, so we think the smartweed, and I saw there was another question about, I referred to pondweed. It is smartweed, not pondweed, but sometimes common names get intermixing. Smartweed, it may have needed a little more soil mass to actually become bigger plant growth. And that's something that we have to just look at its needs specifically. The fact that it's pulling into the soil, that's not a bad thing, but it's potentially would re-release out and we can't harvest it off. So that's the downside of it being stored in the soil, is it's hard to get to. But it may actually, if we can fine tune the growing needs of smartweed, we may be able to use smartweed very effectively, because it can get meters tall. I've seen it up to 3-4 meters tall in water. So it has a lot of potential still, just not on this type of experiment so far.
And Stephen, you touched on it in the beginning of the presentation. Can you talk about the floating mats? Did you purchase the floating mats from a company or were they prepared by your team? Do you want to touch on that again?
So they were purchased from GP Restoration Solutions, which is my secondary company, which actually has the patented commercial floating islands. And we utilize basically a similar type of format to our commercial islands, just in a simpler form. So we used PCV pipe and as a frame with air in it, just to make a cheap, simple floating platform, but it replicates our commercial islands that have a metal casing with then a sprayed on plastics, which can't sink. So it's basically simulating what our commercial ones do, and same growing mediums and functions of the commercial ones, but they don't look as nice as you can see in the photo here that we've got of them throughout the presentation.
What controls can be used and were used to prevent algae bloom in trial 2?
And we did mention that it was mainly shading the water surface, so there's no surface water. So there's no sunlight hitting the water. We were basically making it so that the algae didn't have everything it needed to grow, creating deficiencies. And I will have to check in on if there was some other technique we used, but we didn't want to add something that would kill all the microbial biomass. That was a key consideration because we thought that was important to the function of these islands.
And the next couple of questions are for phase 3, Dan, so I'll ask you to unmute. Are the ponds in phase 3, are they facultative, oxic, or anoxic in nature?
Well, they're in constant flux, right? If there's a lot of inputs, they can be more oxic, but typically if they're sitting there for a while, they're anoxic.
And for phase 3, are you mixing the native plant types on the same floating islands? In the research, it looks like they're all different. So are you mixing them together on floating islands, or are they all staying separate for phase 3?
For that, I'm going to defer to Stephen Tanis because we're still kind of in that plant selection phase.
Yeah. So my preference for sampling is to grow them in monoculture. Now, the islands we use have 2 growing areas in each island that we can have 2 different species growing side by side, but it's really hard to sample the roots of them if they're mixed together. So we can't split the roots apart. So if we're going to analyze the roots, we have to have them separated from each other. So that'll be the consideration, but there is a community thing that benefits. That's why I want multiple species as well.
And for phase 3, Dan, how were the ponds chosen to be either controls or the island sites? How did you guys make that decision?
Well, we wanted a diversity of sites, and so we wanted a control that was a little bit outside of our geography, and we wanted a control that was kind of in the area where the other 3 ponds are. And so we chose Clausen AgriVentures West as the control just east of Linden because it was in between the other 2 ponds. And so it was really reflective of that environment.
Another question, are there seasonal or environmental conditions in which floating islands would be more efficient in nutrient uptake? And how would this affect the experimental design and future applications?
I guess I can take a little bit of that one. Where your plants are growing in the summer is where you get most of your nutrient removal. And when you're using plants exclusively, usually winter is a downtime that you're not having the removal. So you have to design around that. The other aspect that we have is in selenium, we actually had the microbial activity happening all winter long, and we actually had higher drops in selenium in the winter in the ponds with islands and without the islands. So sometimes if our microbial is able to happen underneath the water surface, then you can actually get something to happen in the winter as well. Yeah, generally it's a summer treatment. So you don't want to release the water midwinter.
Another question, is there an opportunity to select species for the floating islands that can be harvested for other uses, perhaps even for small scale livestock feeding purposes?
Yes, that is definitely, we've stuck with plants that are native, but many of these plants are very much edible. If you see any of those photos we had of birds eating them off, a lot of the species we're selecting, sedges are edible, but you'd probably want to throw it through a silaging process because they're not necessarily the most palatable always. But if you put them into a silage mix, they would be definitely high nutrient, but you would do your water testing to make sure that you're just dealing with nutrients and not any metal issues.
And just to play on that, I know we touched on it in the presentation. There's another question. If the wildlife influenced your plant growth, and did they try to eat your plants in the outdoor trials, which I think is a big yes.
Yeah. So when I do outdoor trials, I do control for wildlife, and you'll see a few of those islands that we had in there that had little fences on them. That's for bird exclusion. We have a grading on for muskrats for digging up the roots. In our mine trials, we have not had to do that. We've not had muskrats on our islands. So we leave the grading off, and then it's the birds, when you're talking about 7 centimeters of heat, the birds get their butts wet if they try to nest on them. So they just don't nest on them. So in our mine trials, we've been fine without anything, but when we're working in Lethbridge, we always have a grading and fence on because we've had problems with that before. And I built the island's design to actually integrate a locking and fence system.
And this is an interesting question too, actually. Would it be of benefit to collect data for this next phase on what the cattle are fed at the various sites? Specifically thinking about vitamin, mineral supplementation, protein supplementation, that could be a source of variability of the nutrients that are found in the ponds.
Yeah, that's a great idea and an entire study, I think, in itself.
Phase 4?
Yeah. We are, like I said, we are collecting the samples of the runoff that does enter into these ponds, and we are doing some pretty broad sampling of what's in that. And so it might be useful data for that kind of research down the road.
And can you comment on why it's important to improve water quality within runoff retention systems at confined feeding operations? How do you envision that this technology will improve water quality management at the landscape scale in Alberta?
Well, I think water is going to be one of the most pressing issues in the prairies going forward in the future. And so I think being able to improve the water quality for its potential reuse, right, alone is a big benefit. These are typically contained systems, but if we can use these kind of natural systems to phytoremediate and increase our water supply, I think that's the main benefit.
Perfect. And we have one last question, and I think we have, oh, sorry, Stephen, were you going to jump in on
Yeah,
that?
I'll just add to that because I deal with this a lot, and water supply is a big issue. And these systems are being built to basically handle all the water coming in and evaporate it off. So they basically become a sink for nutrients. So the water gets more and more to the point where it can't be used. And when we're short on water in our prairie systems, if we can clean this water and then use it and then not have as big of holding ponds, if we can do this properly, we may need holding ponds that are a fraction of the size, taking up less space and less cost to build. So there may be actual benefits long-term to future designs.
Perfect. And our last question for our last minute of the webinar, we filled up the whole hour. Dr. Tanis, for plants like smartweed, should they be staked since they seem to be a creepier plant?
Yeah, there is a potential. It likes to creep or to just float in the water, and we didn't give it any water to the sides to float in. And that might have been its problem, as well as not giving anything for it to go up on. And so there might have been some limitations that we provided in its growth that need to be revisited.
Okay, so it looks like we've covered all our questions and it's exactly 12 o'clock. So is there anything else, Stephen or Dan, you want to add quickly? Or I think we've downloaded a lot of content.
No, I don't think on my end.
Okay, so great. Thank you, everyone. Thank you for joining. We had a long list of participants. So thank you for joining. We're going to be adding a recording of this webinar to our website, oldscollege.ca. And please reach out to OCCI if you have any further questions or comments or any feedback. Thank you so much, everyone.